Hi, Abraham. How are you? Thank you for coming. Um, we will start on top of the hour. So yeah, we'll still have a few minutes. And you're muted, by the way, in case you're saying something. Oh, Joyce, I'll invite you. David, I'll invite. Hi, Joyce. How are you today?
I'm good, thanks. How are you all?
Good, good. Thank you. Hi, David. We'll start in
Hey,
around. Hey, nice seeing you here. We'll start in around 6 minutes. So yeah, we'll have a few times. If you think this room is interesting, feel free to share the room in the meantime with people. It's a really interesting new neuroscience tool that will be quite helpful and groundbreaking, I think. So yeah, we'll start soon. In the meantime, check out Dr. Abraham's lab website that I pinned on top of the room. It's really interesting. And in the chat, I will share the paper. It's open source, so you can look at it. I'll do that right now. Hey, welcome everyone. We will start on top of the hour. In the meantime, feel free to check out Dr. Abraham's website, lab website, and the paper that we are discussing is posted in the chat. And I'll switch when we start to the PowerPoint presentation. But in the meantime, feel free to check it out and invite people that you think that might like this room here. Thank you. Okay, we are almost about to start in 1 minute. So, Ibrahim, do you want to test out how your sound, how you sound? Can you hear us? You're muted, just in case you. In case you. Okay. Maybe you have to come back in, like leave and restart the app and come back in if the sound doesn't work for you, if you can speak. And okay, I think we have some technical. Oh, okay. Please. Okay. We will be fixing this in a minute. If anyone else has advice, usually it's either that universities block social media apps, or the first time people use the app sometimes has technical issues, so restarting the app usually should fix it. But if anyone else has have had similar issues and has advice, feel free to add it to the chat, and we will start as soon as we figure this out. Yeah, I don't know, Joyce, what we can do right now. Do you think moving Abram to the audience and then bringing him back up helps?
Couldn't hurt. I don't have any idea though. Or even him leaving the room and coming back. I don't know.
Okay, I'll switch to the presentation in the meantime so we can try out if the presentation is shared with everyone to view. If that doesn't work, I have a solution for that. So Joyce, can you click on the presentation and check if it's working? Thank you.
Okay, I will check. It looks like it. Yeah, looks fine. Do you think it would help to make him a moderator?
I don't know. He's not restarting the app. Oh, okay. So, okay. Let's hope it just works. Hi, Jake. I see you raised your hand. Thank you for coming. Okay, let's see if Abraham's can you hear us, Abraham?
Hi, Abraham, if you can hear me. Well, I guess he can't hear me, but I guess if you can message him and just tell him to leave the room and then come back in,
Yeah, he did that.
I think it will fix
I
it.
think it's the problem is he is on university Wi-Fi and it's blocking. We had that issue before.
It's an issue I've seen in the past couple of days in several rooms.
Okay. Yeah, I hope he is able to switch to cellular data. If not, I will try my best to in the meantime talk about the research. It's really so. I don't know if I take away too much, but in general, it's really important to know to get more insight into dopamine circuitries in the brain, and okay. Okay. It works. He switched to a phone and still no luck. And I hope he... Yes, now it works. Can you hear us?
Oh yes, I can hear you now. But I'm on
Perfect.
my iPhone though. I don't know how effective.
Yeah, it's perfect because now we can hear you. And since this is not the screen
This
share,
medium before, and I'm totally new to this, so my apologies.
yeah, don't worry about it. It's fine. Since this is not the screen share, people have to go on the presentation link themselves and scroll through it. There's really no need to use the computer and the university Wi-Fi. We had this issue before, so you're not the first one. So thank you for coming. And yeah, let me give the audience a short introduction and then we'll go from there. So welcome everyone to Science Society, and of course a special welcome here to Abraham. We are so glad you made it and that you stayed persistent, although we had some technical issues, so thank you so much. And Dr. Abraham Beyene, he's a group leader at the HHMI's Janelia Research Group research campus institution, and he is interested in developing new nanoscale optical probes to study neurotransmission. And he and his team focus primarily on probes for chemical signaling molecules, including neurotransmitters, neuromodulators, and neurohormones. And the group uses tools of material chemistry to create new ways to image and sense the shortwave infrared part of the spectrum. And why? It's because it has remarkable tissue penetration, which is really important and useful. So welcome to Science Society. Thank you so much for being here. And we usually start off with a couple of interview questions, if that's okay with you.
Sure, but just as a warning, I really have like not a good connection. I'll try my best to stay on and go through this, but let's go ahead. I'm hoping to get lucky, but
so far we hear you really well. I know sometimes we have the red bar sign, but nevertheless, usually it works fine. So
okay.
let's hope for the best. Okay, so Abram, thank you so much for coming again. And do you know, like we usually like to ask this question our scientists that come here. What was how did you choose the path of science? Like, was it something you always wanted to do? Was it a teacher, a book, maybe something that happened that kind of sparked your interest in science? Thank you.
Well, thank you for that question. Yeah, so I growing up, I've always been I would say like a good student. I really loved school. Basically, I loved reading. Uh, when kids complained about homework, I actually really enjoyed it. I don't know why, but I guess that was just the way I was as a child. I was just really um, attracted to learning and education and so on and so forth. But the idea of like going on and becoming a scientist was still not something that I thought was possible for me, because it wasn't clear to me what the path was, right? So, and I grew up in a family that was supportive, but I did not have people with advanced degrees around me that told me, to become a scientist or to have a career in science, you have to go to graduate school and get a PhD and so on and so forth, right? Which I think when you reflect back on the journey that I had, seems like a very straightforward journey where people go from undergrad to graduate school, get your PhD, and then go on to pursue science. But if you don't have the familial and social support around you as you grow up, you don't necessarily see that path. So for me, the turning point was going to a school. I went to my undergrad was at University of Maryland, Baltimore County. Uh, so that school had this program, the Meyer of Scholars program. I don't know if you guys have heard of this before. And the Scholars program supports people like me that do really well in undergrad, but not only do they do that, they also encourage those students to go on and pursue advanced degrees. And that was like in that undergraduate phase is when I began to see the path. So, so you have to prepare for graduate school, you have to do undergraduate research. It's not just about the grades, but you have to have some kind of undergraduate research experience under your belt. All of these things they tell you, and then I was really basically very lucky to have been part of that program that ended up giving me this opportunity to pursue this career, I would say.
Yeah, I really appreciate hearing that you went to a school that really supported you in that path. But I would frame it differently. We are very lucky that you took that opportunity because what you're developing is really important for neuroscience. So I think we are the lucky ones, not the other way around. And how did you then choose this path of creating these amazing tools?
Yeah, so even that is a bit of a fortuitous coincidence. So I did my graduate school at UC Berkeley in the chemical engineering department. I was really attracted to the physical sciences more than biology. Um, but I found a PI. She was still a postdoc back then. She had just gotten a job as an assistant professor at Berkeley. And she was still at MIT, and I reached out to her and said, you know, you're not here. You're still going to be like another semester or so before you come here, but I really want to be your student because I'm interested in nanotechnology. So that was my interest primarily, was to really look at the materials chemistry aspect of nanoscale materials and study them from like a physical sciences perspective. So anyway, so yeah, I became her student, and we began to characterize these really interesting nanoparticles that I'm going to be talking about today. And then found out that these materials have a really unique photophysical sensitivity to a class of neurochemicals in the brain that we call catecholamines. So that ended up taking, I would say, the second half of my PhD thesis into the neuroscience role area and into the neuroscience research area. So that's how I ended up going from basically working on nanomaterials to then straddling the boundary and going into neuroscience and neurobiology and exploring the utility of these nanoparticles and these nanoscale materials for studying uh, things that are relevant in neuroscience, particularly on this molecular and cellular neuroscience side of the neuroscience research. So, and again, I don't think I had like a grand vision or plan ahead of time to work on in this area of research. I would say that I just sort of followed what was immediate. So basically, the way I went about it is I'm going to do as well as I can on the projects that are immediately in front of me, and then just let that take me, let that guide me, and see where that takes me. And that is how I ended up where I am right now. So I wish I can give you like a very big and grand vision of how I got to where I am, but it's really more of a series of decisions and that took me to where I am now.
Well, I'm glad that everything worked out, and I don't think all grand visions always work. I think it's really interesting and always delightful to hear that people just follow curiosity and try to do their best, and something beautiful like this comes out of it. That's an amazing story, and yeah. So yeah, the presentation is pinned on top of the room. The stage is yours. So we are all curious to hear more about your research. So thank you, and yeah, please, the stage is yours.
Okay, well, so how I'm assuming that everyone has a link and they would be able to follow me. Is that how it works? So if I were to begin to go through these slides, people would be able to follow what I'm looking at and what I'm talking about.
Yes,
Is that right,
exactly.
Katarina?
Yeah, everyone can click on it, and it's really helpful when you say when you switch slides that you mention it. That's all. But yeah, thank you.
Okay, sure. So first slide, then let's just get right into it. On the first slide, if you're not in animation mode and you're just looking at the whole thing, you will see some cartoons. And I want you to look at the cartoon on the top right-hand side that shows these nanoparticles that we use in the lab. That's going to be sort of the star of the show for today. It's called a single-wall carbon nanotube. It's a really interesting nanoscale material. And the best way I can describe nanotubes is to actually really go back to the parent graphene sheet. Graphene itself is a really fascinating nanomaterial in its own. It is a two-dimensional nanomaterial that is made entirely out of carbon. And graphene is made out of this single carbon atom thick sheet, right? And each one of these dots or vertices that you see represents a carbon atom that is bonded to 3 other carbon atoms through this so-called sp2 hybridized carbon-carbon bonds. So for that reason, it has this hexagonal planar structure that sort of extends in 2 dimensions. Now, if you think about rolling that graphene sheet into a really tiny and hollow cylinder, you get what are known as single-wall carbon nanotubes, or just nanotubes for short. So one nanomaterial, when you roll it and change the geometry of it, ends up giving you another nanomaterial. And what is really striking is because of this extreme curvature in the radial dimension for the nanotube, which is on the scale of a nanometer, the properties of nanotubes themselves are really different and distinct from the parent graphene sheet. And there are two properties that I want to highlight today that's going to be relevant for my talk. And the first one is the fact that these nanomaterials are fluorescent, and they fluoresce in this range of the spectrum that we call NIR or SWIR. So here we're looking at on the chart or the graphic on the bottom left corner. So what you see there, if I draw your attention to the x-axis there, so you see that emission happens in this 850 to 1350 nanometer range. And that what you see there is a fluorescence emission spectrum. So you can see that it is a fairly complex looking spectrum with multiple peaks all confined within that window. And those peaks arise from slight differences in diameter that could end up having slight differences in emission. In any case, what I want you to take away from this is the fact that these nanomaterials, unlike graphene, have their intrinsic fluorescence to them. And then the second takeaway from this slide is that their fluorescence is very photostable. So if you look at the red trace on the right bottom chart, that is a fluorescence emission from a nanotube dispersion that more or less puts out the same fluorescence output after hours-long exposure to the excitation light source. And that is in contrast.
Sorry, we can't
So
hear
a
you
lot of
right now.
organic fluorophores and fluorescent proteins that are.
Oh, you broke up for like 20 seconds or so. That's all. Sorry.
Okay. Anyway, it's okay. So I'll summarize this slide. So the summary from this slide is that these are fluorescent. They fluoresce in this near-infrared region of the spectrum, and they're also extremely photostable, meaning that they don't bleach.
I'm sorry, you broke
Here
up
is
again.
a strategy that enables us to make sensors out of these nanomaterials. Okay, and the way you can make a sensor out of
I'm sorry, you're breaking up a lot. Did you maybe move to a different place?
that are in close proximity to that hybrid material. Okay, that
Yeah, I'm sorry.
detection,
You're breaking up a lot.
supermix shift, that could help you. It's nanomaterial, meaning that you can have a fluorescence. So
Abraham, can you hear me? We okay, I don't know. Is it just me or can anyone hear?
No, I can't hear him either. I didn't hear most of what he said after he started talking about how we create a sensor out of the nanotube.
Okay.
Yeah,
Did
I'm
you
sorry, guys. I don't know what we can do.
did you move maybe to a different spot? In the beginning it was just fine, just when like the last couple of minutes or 3 minutes or so was then breaking up.
Yeah, it's yeah, I'm trying different areas here, but no luck so far.
So right now you're fine.
Yeah, okay. Well, let's try this. Let's
Yeah.
give it a try.
Okay.
And if this becomes an issue, then we can really just find some other ways of communicating this. But in any case, yeah, let me know if I'm you can't hear me, but I'm gonna keep going right now. So I was just describing on slide 3 how one makes a sensor out of these nanomaterials. And the takeaway from this is that the material can be coupled in a way to create this other hybrid nanomaterial that has a chemical matter that is decorating the surface, and somehow that decoration on the surface could end up recognizing molecules that change the fluorescence properties of the nanomaterial, and that can be used as a way to read out that molecular recognition. Okay. Can you hear me?
Yeah, right now it's perfect. Thank you.
Okay, very good. So next slide then, slide 4. That takes me to the topic of today's discussion, which is these nanomaterial based sensors for a class of neurochemicals that in the brain that we call catecholamines. So this slide is highly animated, so you might want to try having this in animation and just tab through the whole thing to bring everything up. So I'm going to begin with the top graphic there. So what you see there is a pristine nanotube that has not been conjugated to anything. It's just a pristine nanomaterial. And imagine that pristine nanomaterial has some kind of intrinsic fluorescence that we can call F0. And then these nanomaterials, like I said, are extremely hydrophobic because they're made entirely out of carbon. And for that reason, to actually be able to access their photophysical properties and to use them in any meaningful way, you have to decorate the surface. You have to really functionalize it, elaborate the surface with different types of chemical matter. You can do this covalently or non-covalently. And it turns out that one of the chemicals that you can assemble on the surface of these nanomaterials is actually single-strand DNA, right? Single-strand DNA creates a really stable colloidal dispersions out of these nanomaterials because it has an amphiphilic property, meaning that it has a negative charge in the form of a phosphate backbone, and also has these hydrophobic nucleobases that can pi stack onto the hydrophobic surface of the nanomaterial, and thereby stabilizing it and keeping it suspended in solution. But what is interesting is DNA, when it decorates the surface, ends up quenching the intrinsic. You can
Just
hear me,
lost you again
correct?
just now.
On my end, it's not muted. Everything seems okay.
What's weird is your mic appears, the icon appears to be muted, but we can hear you. But you just dropped out for the past like 10 seconds or so.
Okay, very good. Okay. All right. So,
But can I ask you a question if I understand this correctly?
this DNA.
So the nanotube, the fluorescent properties of the nanotube changes based on the environmental chemistry or something, right?
That's correct. Yes. Yes. That's precisely. So that's yeah. That's a one sentence summary of what I'm talking about on this slide, which is that if you take the DNA on the surface, it ends up quenching the intrinsic fluorescence of the nanotube, and then that quenched fluorescence can be rescued by the addition of these neurochemicals that we call catecholamines. In this particular case, I'm showing a dopamine here. So dopamine can come in and turn up or reverse this quenching that is caused by the DNA on the surface. So the is that the local chemical environment really has a very profound effect on the photophysical outputs of these nanomaterials, and that is what we're trying to leverage for sensing purposes. So can everyone hear me?
Yeah, I can hear you right now. Thank you.
Very good. Okay. So the graphic on the right-hand side of this slide shows this fluorescence perturbation caused by dopamine. So this is a sensor in action. So the black trace there is the nanosensor without dopamine. And then when we add dopamine, you can see that the fluorescence goes up, it increases as a consequence of the addition of dopamine. Okay. And what we also know is that this turn-on event is a reversible process, meaning that if you were to take the dopamine away, this thing is going to get quenched. It's going to go back down to baseline. And then when you titrate the concentration of dopamine and compute a dissociation constant for this system, you find a value that is on the order of about 250 nanomolars. So At a very high level, this is what the sensor looks like. So I'm hoping that you hear me, but if I try to summarize everything that I've told you so far, it is this. So if we take this inorganic nanoparticle in the form of a nanotube, and then conjugate that to DNA, which happens to be one of the central molecules in all of biology, creating this hybrid nanomaterial. That hybrid nanomaterial has a functionality that is really distinct and unique and different from its unique ability to respond vigorously to the addition of dopamine. So the presence of dopamine in the local chemical environment of this nanomaterial ends up creating a very strong perturbation of the photophysical output of that nanomaterial, and that we can use that for imaging dopamine. Right? We can take advantage of this thing that we observe in a test tube, and then deploy it in real biological specimens to study biology. So that is a takeaway from what I've told you so far. Is everyone able to hear me?
Yes. Right now we're good. Thank you.
Very good. Okay, so let's go to the next slide then. So this is again, this slide is highly animated, so you can tap through the whole thing and just bring everything up so that I can explain.
Slide number?
This is slide number 5 now.
Okay, thank you.
Okay. So this slide number 5 basically has this title that says nanotubes can be deployed in unconventional but useful ways. So I'm reading this just to make sure that we're all on the same slide and looking at the same figures. So what you have on the right, on the left top corner, is this composite nanomaterial that we've been able to make out of these nanosensors that has enabled us to visualize synaptically resolved effluxes of this neurochemical that we call dopamine. And to explain this system, it's actually a very easy one to understand. The very bottom layer of this composite material is glass. So you just think of a glass cover slip, right? And what we've done is we can use some very simple chemistry to functionalize the glass so that it has a positive charge on the surface. And then because the DNA itself is negatively charged, we can create a very stable nanofilm that sits on the glass and is attached or attracted to it through electrostatic. In creating this composite nanomaterial, and then you can culture neurons on top of this. And what we've done here is we've cultured primary dopaminergic neurons, and the hope is that if we stimulate some of these neurons that are growing on this substrate that is dopamine sensitive, we might be able to capture local releases of dopamine as this released molecule goes and activates some small subset of sensors that are in close proximity to where this release is occurring. So the goal here is to be able to image at a really high spatial resolution that enables you to localize specific activities to chemical synapses of dopamine. So on the left left bottom, what you see here is a scanning electron micrograph of a dopamine axon. You see these axonal swellings. So these things that look like beads on a string, those are called boutons or varicosities of dopamine neurons. And those are the places we expect to see dopamine release. So you can think of those as chemical synapses of dopamine. And the hope is if dopamine gets released from some subset of these boutons, and might be able to activate the medium on which it is growing or on which it is sitting so that you can capture activity. So that was a vision. And then the movie that you see in the bottom right-hand corner is after multiple rounds of optimizing the system, we've been able to get it to work really well. And this is one of the videos that we've been able to produce where I'm showing you a single process of a dopamine neuron. And you can see this dopamine that are getting released from specific loci across this particular process, and then that released molecule then goes on to modulate the fluorescence of the medium on which this process sits, and turns it transiently, so you can capture that transient turn-on event. Okay, so that is effectively a kind of like a proof of concept demonstration of this system. Is anyone able to hear me?
Yeah, I can hear you fine.
No, okay, no connectivity issues. Okay, very good, very good. So, all right, so this is what it looks like. So let's go to the next slide. So slide number 6 is effectively the same thing. So David has a question here. Is this a form of amplitude modulation? Because there also seems to be a frequency shifting property. That's very good, David. So this is an intensity amplitude modulation. That's precisely correct. There are some sensors that are based on what you call frequency shifting or wavelength shifting. But in this particular case, what we see is that the amplitude or the brightness of the sensor just goes up, and it really doesn't change the peak positions of these photo emissions. All right, okay. So let's move on. So what you see here in this video is effectively a bigger version of the video that I shared earlier, where we're seeing release, a synchronous release from hundreds of release sites, sort of simultaneously releasing dopamine onto the substrate on which they're growing, and in so doing, they end up modulating and turning on the fluorescence of the media on which they're growing, and we're able to capture that here in this video. Okay. If you have any questions, please feel free to jump in. I'm happy to have this sort of be like a back and forth discussion. If there is anything that is not clear, I'm happy to elaborate more and explain things better.
Could I ask?
This
okay.
is yeah.
Okay, go ahead, TJ. I'll come
Yeah,
up.
just a very quick question, really. Fascinating, not my field of expertise at all, but very interested in this kind of research. Could you give us a couple of examples? Or I know it's in R&D or you're doing the research on it, but have you visualized, or is there any applications where this is being used? Apart
yes,
from the
so
ones you've
why don't I
described
go
here, for example.
Abraham, we cannot hear you if you're there.
I think maybe you muted yourself by accident. Can you hear us? Oh, yeah, now you're unmuted.
Okay, so you can hear me, correct?
Yeah, now I can hear you. Yep, thank you.
All right, very good. Okay, so let's go to slide number 7 here. Here again, you see a highly animated slide, and I want you to tab through everything and just bring everything up so that we can look at it together. Here, the takeaway from this slide, it looks very busy, but the takeaway from this slide is that if you take a closer look at these videos, so the motion picture, the video that I showed you earlier, they're all very interesting and nice to look at, but what kind of information do they contain? Like, is there any kind of utility there in going about imaging in the manner that we've just done? And I'm trying to answer that here. And what I'm showing you here on the top row is a sequence of frames from the video that I just shared in the previous slide, where at the moment of stimulation of this field of view of imaging, so that refers to the STEM image, you see these blobs of activity or clusters of pixels that are highly correlated that emerge at the same location across stimuli. So what I mean by that is if you stimulate several times in this field of view and just take a look at the video at the frame at that immediate position of stimulation, you see the emergence of these same hotspots of activity occurring at the same location, having more or less the same shape. And then about a second later, these hotspots sort of blend into one another because dopamine is diffusing. So you get this semi-homogeneous looking field of view. And then after some period of time has elapsed, that turned on sensor goes back to baseline. So this is a reversible sensor. And then if you average activity across multiple stimulations, you see that you get that temporal trace that I have on that top right hand side. Okay, let me pause here. There is a question. For clarity purpose, what type of neurons can be used? iPSC derived neurons versus in vivo neurons. Oh, that's a really good question. So these are primary neurons derived from in vivo. These are collected from rats. We culture them and then we image them. And we have a project actually right now going in the lab to try and do this with iPSC derived dopaminergic neurons. And for those for those that may not know what iPSC is, iPSC stands for induced pluripotent stem cell. Effectively, it could be like a human-derived cell that is then made pluripotent and then differentiated to become dopaminergic, and then can we image, right? Can we image from that from such a system? And we do have a work in the lab that seeks to do precisely that. So that's a question from Ilya. I hope I've been able to answer it. Okay. Now let's look at the bottom row together. So at the bottom row, what you have is just a bigger version of the stimulation field of view. And what I've done there is I've brought up another channel that has THGFP on it. So what I mean by that is it's just a structural image of the neuron that is giving rise to this activity. So you can think of the boutons or varicosities that I talked about earlier. So those are the anticipated sources of dopamine. And then we overlay this hotspots of activity with the GFP image to convince ourselves that these activities that we see here are truly being driven by effluxes of dopamine from specific boutons or specific chemical synapses of dopamine neurons. Okay, that's what you see there at the bottom. So the takeaway here is that what you see here as clusters of correlated pixels that form as blobs or hotspots are truly effluxes of dopamine that are being captured from specific chemical synapses of dopamine. So this is something that has never been done before, but we're doing it here. We're capturing that very early phases of escape of dopamine from the release side. And not only can we visualize that, we can also co-localize that activity to specific boutons or specific chemical synapses of dopamine neurons. Now, on the slide number 8, what I have there is these biosensors in neuroscience that are very extensively used. We have calcium sensors, voltage sensors, and neurotransmitter sensors. And if you tab through this slide and bring everything up, the point that I was trying to make here is that most of these biosensors, in the way they're deployed, emphasize collecting activity from as large a number of neurons as possible, and it is really rarely targeted at the subcellular level. So the type of imaging modality that we're advocating for in this study really drives home the importance of looking at things at a subcellular level, like looking at things not at a population of neuronal level, but going subneuronal, subcellular, and looking at things at a single chemical synapses. So there are a couple of questions that have been brought up. So let me read them and then try to answer them. So this is from David, and it says, is there important information in the phase response of pixels which end up fluorescing? Do the parts of image that end up the brightest stuff start? Oh, that's really interesting, David. I think to be able to utilize this information, we would need to do the type of imaging that we're doing, but at a faster frame rate. Right now, I did not share this detail, but the speed with which we're imaging is approximately 10 frames per second or 10 hertz, and although we do see some slight differences in in terms of how quickly these clusters of pixels attain their peak fluorescence, that difference is usually just a frame or two. So it's really not, we don't have that temporal resolution to really make that determination. But I imagine that information could be useful if you're imaging at faster frame rates, something like 25 hertz or 50 hertz imaging. And then I think at that point you might be able to, you may be in a position to try to extract some useful information from that. Is everyone hearing me? So I'm not sure if people hear me or I'm just talking into. Okay, very good. And then Ilya had another question. Are we limited to dopamine neurons? Could other types of neurons be used? So that's a really good point. And again, I think that brings up a question of, can we make sensors that are for glutamate or for serotonin or so and so on and so forth? Right now, the answer is partially yes, we can do it for other types of neurons, but not all types of neurons. The reason being that we don't have sensors that target these other types of neurochemicals. Primarily what we have right now are catecholamine sensors, which is good for dopamine and norepinephrine. And we also have serotonin sensors. So we can do this with serotonergic neurons, which are fascinating. But right now we can't. I would love to do this on glutamatergic neurons, but we can't do that right now. Okay.
What about GABAergic?
And not GABAergic, no. Not glutamatergic, not GABAergic. But also I think the way I approach questions in my lab is there is half of it is dedicated to biology and the other half is dedicated to the physical sciences, like the chemistry aspect of this. And in that half of the projects, we try to answer this precise question, which is how we make sensors that are similar to this, but for other neurochemicals like glutamate, GABA, acetylcholine, and so on and so forth. So Jared has a question, which is, how is this sensor different from something like Neuralink? I'm not familiar with Neuralink, but I imagine that these neuropixel probes and Neuralink probes probably rely on detecting electrical activity from neurons. Here, what we're trying to do is actually detect chemical signaling from neurons. So neurochemical signaling is very important because once an electrical activity in the form of action potential invades a chemical synapse, it elicits a chemical release. So communication between neurons is really some mix of chemical and electrical. So here we're focusing on the chemical aspect of things, but this Neuralink sounds like something of an electrode-based technique that measures electrical activity from neurons. Again, a very important piece of technology, but a different modality of sensing. All right, let's go to slide number 9, guys, and tap through this all the way through. Not all the way. Stop before the last animation. All right, so what
Is it just me, or is his signal going in and out?
It's his signal. We cannot
Oh,
hear.
I think, Abraham, I think you muted yourself by accident. I see the mute button up. Can you hear us?
Potentially not. That might also indicate that. Oh, there we go.
Okay.
did I drop off?
Yeah, I think you muted yourself by accident. The mute button was on.
So
Okay.
you said let's switch to slide, I think 9, and then we lost you.
Yep.
Oh, I see. Okay. All right. So, yeah, I'm on slide 9. I was just talking about what I was trying to communicate here. So if you tab through these animations on slide number 9, go through all the animations except the last one. So stop before the last animation. Yeah, so I'm still here, right? You can still hear me?
Yes.
Very good. So very quickly, what you see here is there are 5 activities, 5 blobs or clusters of activity on the NIR DFFRF image, and then on the THGFP image, you see the varicosities or the chemical synapses of dopamine that are presumably responsible for giving rise to these clusters of activities. Okay, so what I'm showing you here is that for each one of these clusters of activities, I can assign them to specific varicosities, specific chemical synapses that give rise to those activities, right? And I can do that because I have an overlay image that I can use as a guide. What is really striking is that when we image,
Is he gone again, or is it just me?
Yeah, I think there's an issue with connectivity on his side.
Yeah, Abraham. And now you're muted again. Sorry. In between it was fine, but right now you're breaking up, and you're muted.
The mute tends to be an auto function of the breakup, unfortunately.
Oh, okay. I didn't know that. Probably you can't hear us right now.
Probably not. We'll see if it comes
Yeah.
back again. It happened.
Now you're back.
okay,
Perfect.
how much was I able to communicate this time around?
Well, you said we should stop before the last animation.
I see.
Yep.
Okay. All right. So I'm still here, right?
Yes. Now you're here.
All right.
Abraham, is there potentially any way you could move closer to a wireless router, or maybe if you're on GSM, switch to Wi-Fi or anything of that sort?
Well, I was on Wi-Fi, but I think Katarina told me I need to be on cellular data to be able
Yeah. Are
to
you
do
still
this. Correct?
university Wi-Fi? I think the problem is with university, like most universities block social media, so Abraham will look like he's here, but he won't be. He didn't hear us or could speak, so that's why usually just getting off the university Wi-Fi helps. But yeah.
Yeah.
Or have you
mm,
tried? Maybe it's different where you are. I don't know.
Well, that was the beginning problem, like the first 6 minutes or so was trying to fix that. And
I'm back on a. I tried a different Wi-Fi connection now, so maybe this would work.
yeah, we can hear you right now. So yeah, it should work then. Thank you.
All right, so let's go ahead. So animate to the last one. Just stop before you go to the last one. So so in this field of view, you see this NIRD of a VRF image, and then you have the THGFP structural image and the overlay image. In the NIRD of a VRF image, this is activity images, and there are 5 clusters of pixels that are sort of like the putative releases from specific varicosities, right? And because I have that overlay image, I can determine with a great degree of confidence precisely which varicosity actually leads to which activity, right? So these white arrows are meant to guide you with that effort, where I can say, the one of these varicosities gives rise to one activity image, and so on and so forth. What is really striking is when we image in this manner, um, the there is a subset of chemical synapses of dopamine or varicosities that actually do not contribute to release. Those are depicted in white arrows here. So the white arrows, you can see if you look at the corresponding NRDF image, you actually don't see any kind of activity from them. So why is that? These white, I'm sorry, the red arrows, I mean, the red arrows show no activity, no corresponding activity. And as far as we can tell, these appear to be chemical synapses of dopamine. At least morphologically, they satisfy the criteria for a chemical synapse, but they really don't have the corresponding NR-DFF activity. And I think this is a kind of information that question that we want to pursue here. So what are some of the molecular level differences between these release capable and release incapable chemical synapses of dopamine? Like what are some of the protein level differences or transcript level differences? And even more importantly, what is a regulatory governing principle here that enables specific sets of varicosities to be release capable, whereas some other subset remains release incompetent, right? So these are, I think this is a kind of information these subcellularly resolved imaging would be able, would be able to help you go after, or would be able to help you investigate. So the last animation basically shows you that even in clusters, we're still on slide 9, the last animation. Even in clusters that have a set of chemical synapses of dopamine right next to each other, you can see that there's 3 of them here. We can conclude with a pretty high degree of confidence which activity actually, which varicosity gives rise to the activity in question. So this is a very important capability that I think this tool is able to provide. So slide 10, at a very high level, the takeaway from this slide is another application of this technology where if you animate all of the slide 10, what you see here is a dopaminergic neuron, in the field of view. And there is a single process that I've highlighted there in red box. And there, if you take a closer look using the bottom row images, you see that this process is co-localized with these 3 other NR-DFF activity images. And this is very unique and very interesting because what we're imaging here are dendrites of dopamine neurons. And in general, release from dendrites is not something that is common. Axons are release capable, but not dendrites. And here, the ability to really do things at a subcellular level has contributed to this this unique capability for us to be able to image dendritic release and then go about characterizing it in a way that has not been possible before this technology was introduced. So go to slide 10. We're nearing the end, but I just wanted to highlight here, this is again a pretty busy slide, but the takeaway message from this is that once we're done imaging in this modality, we can fix these neurons, and then we can go look for specific types of proteins in these processes that are giving rise to these activities. So in this particular case, we went and stained for a protein called MAP2. So that stands for microtubule associated protein 2. That is enriched in dendrites, but it is absent in axons. And the whole effort here is to really prove that these activities that you see here are not axonal in nature, but are dendritic in nature, and really just contributes to that base of knowledge we have now about release of dopamine neurons from dendrites of dopamine neurons. The last slide, slide number 12, also has an interesting dynamics there. If you animate that, you see a segment of a dendrite of a dopamine neuron undergoing this dopamine release that is totally stochastic. So far, what I've shown you is where we're stimulating the neuron. We're forcing the neuron to fire action potential using optogenetics, but in this case, we're not doing anything to it. We're just watching it passively. And this guy just goes and begins to engage in this really vigorous stochastic activity event that is, I think, really striking. And we can take a look at these things. We can programmatically determine these clusters of pixels that are correlated, and we can color code them. We can determine the spatial as well as temporal behavior of these activities from specific processes or specific segments of neurons in a way that, again, is really difficult to do with existing tools and technologies. Slide The next slide, I guess this is slide 11 now. No, slide 13. If you animate that, what you see there are two videos. One is one is an ACSF. So this is our normal imaging buffer. So you don't have to worry about what ACSF is, but think of it as our normal imaging buffer, or the buffer through which in which these neurons are grown, and we can image through them. And then we can also image in that same exact field of view with in a drug called TTX. So TTX stands for tetrodotoxin, and it is a sodium channel blocker. And what it does is it blockades this action potential dependent synchronous releases of dopamine, and then leaves behind these stochastic release events that are known as TTX minis. And that really demonstrates the level of sensitivity that we have from these sensors. This effectively tells us that you can detect the smallest bits of information that these neurons can release. So single vesicle fusion events can now be detected in a way that is resolved in both the temporal as well as spatial domain. And that is, I think, a really unique capability of this tool. Slide 14, let's, in interest of time, let's skip 14. I think there is really fascinating information there, but in interest of time, let's go to slide 15. And then I'll conclude on slide 15, and I'll stop there. And what I wanted to share with you here is there are some challenges with this technology. Now, I told you about this really unique fluorescence emission spectrum at the beginning of my presentation. That emission spectrum goes from F50 to 1350. So that's one of the figures on this slide shows that. And it turns out that this unique range of the spectrum is generally incompatible with a lot of the imaging technology that are currently available in a lot of neuroscience labs, right? The reason that is the case is because most fluorescence or fluorophores, right, protein-based or synthetic small molecule dyes, they're all they all release in this visible range of the spectrum, where silicon-based detectors do a really good job of detecting them. So most of the cameras, SICCD or PMT photocathode materials, including bialkali, multialkali, and GASP, they don't do a good job of imaging in that shortwave infrared region of the spectrum, or NIR region of the spectrum. So for my lab, the first year and a half of it was really dedicated to developing this custom microscopy solution that enables us to image in this very broad range of the spectrum, 400 to 1400 nanometers, meaning that I can image from a green fluorescent protein, or GFP, as effectively as I can image from these nanotubes that emit in this range of the spectrum. So I'll leave it there. I'll stop there and then acknowledge a few people in my lab. John Dima, who is a postdoc in my lab, that was the point person for this project. Deepika from a cell culture facility. Dmitry from our core facility here for his contributions to developing this custom microscopy solution that I talked about earlier. And Casper and Anastasia also at Janelia contributed a great deal to this. I hope that I'm still being heard.
Yep.
Okay, very good. So I don't know. So we've gone for about an hour now, and that's probably because there was a lot of pauses and repetitions. But yeah, if there is an appetite for a few more questions, I'm happy to entertain
Yeah,
them.
thank you. Thank you so much for this really wonderful presentation and this beautiful animations. And would this also be possible to use in slice electrophysiology? Because in the beginning you mentioned that it has to be mounted on specific coated material.
Oh no, yeah. So that's a good point, Katarina. So this is just one specific presentation in that modality that I shared, which is to create this composite nanofilm. But these nanosensors are in solution, so we can apply them to brain slices. We have done that. In fact, we can incubate a brain slice, like an acute brain slice, in this sensor, and then just the passive room temperature incubation for 15 minutes is enough to really good to give you a very good labeling, and then you can go and image dopamine in the striatum, for example. We've done that.
Wow, that's perfect. That's
Yeah.
amazing. And especially amazing that you could also detect like this tonic or minis. I think that's where you will be able to detect, let's say you make a mouse cocaine dependent or something, and then I think you will probably see a lot of differences in this tonic release, which is really exciting. And also, it's really exciting that you moved far away from the spectra that we usually use. I know it's kind of probably for some labs, but it depends. If you don't want to, I mean, this is 2 photon, right? So, but you can just have different filter cubes and an image, separate from your the
Mhm.
indicators that is a
Yeah.
lab or something.
No, I agree. So this, yeah, so that far near infrared, short of infrared, so we're so far out that we can multiplex this with like green channel or red channel activity indicators like calcium or whatever, and yeah, and even try to do that simultaneously, not even swapping between channels, but try and do this in a way that relays information in both channels simultaneously. So, yeah, really very good point, and definitely something that we're interested in doing.
Yeah, it's really cool. Like I bought back in time from Andor like an imaging system where you didn't have to really switch. You have to buy it with 2 cameras, and then you can simultaneously detect in a cheaper way than 2 photon. That also should work with that. That's really great. And you can have you can image calcium and dopamine at the same time, which is really so useful. Are you
Hmm.
planning maybe developing a potassium indicator by any chance?
Potassium, no, that's not something that I've thought about actually. These metallic ion indicators for sodium or potassium. Is there an interest in that?
Yeah, you know that astrocyte research is gaining more traction, and it's really important, I think, for a lot of neurodevelopmental and degeneration, and also for like connectivity and how actually memory processes work, like that astrocytes and so on are considered. And there came a paper out, and we had the guest speaker here, that basically the astrocyte or glia activity starts with detection basically of potassium released from neurons, and then like their whole calcium waves and so on start. So having a really good potassium indicator would, I think, solve a lot of answers that would tell us how like neurons and glia work together, and what the involvement is there.
Hmm. Oh, most fascinating. Okay. Um. Yeah, I have to think about this. I know the literature does demonstrate some nanotube based metal, like just metal, like M plus detection capability. I don't know what the selectivity for these things are for potassium over some other monovalent metal ion. So this sounds fascinating. I don't know. This maybe something that we can
Yeah,
try to do. Yeah.
and then you would be out trumping at Biden. He said it's really difficult right now and so on. He's coming here to Science Society in November. I don't know if you want to come too. You guys can maybe chat, but he didn't manage until now to create one, so that would be amazing if you could.
Yeah. And who was that, by the way? Who was a
Oh,
person?
Ed Boyden, He's at MIT.
Ed Boyden. Okay.
Like developed a lot of like neuroimaging.
Oh no, I know him. I
Oh,
know
okay.
Ed Boyden. Yeah. Okay. Cool. Yeah, that sounds really fun. Yeah, I think if anyone can do it, I think Ed's group might be able to pull it off.
I don't
They're
know. Maybe. I mean, they're also focusing on
they're
other stuff maybe right now. So
yeah. No, but he does have the bandwidth, the people to be able to do that kind of stuff. Yeah.
yeah, Ilya, go ahead.
Could I? Yeah, I had a question. In terms of practical use for it, so I'm working on gene therapy with Jutsu Molsky. It's a nonprofit effort, and we have IPS lines from the patient, and then we have the isogenic IPS lines from the same patient, but the mutation is CRISPR corrected. Disease is neurological, and so the point that I'm trying to get to, could this be used to show the rescue phenotype? We know that dopamine neurons are affected. We know that serotonin neurons are affected, GABA are affected, right? And we can understand what the how it should look like because we can create the isogenic control neurons, and so compare them side by side. So what would be the practical use of this?
Yeah, I think my so I would have to, I guess, look at the background on this and sort of familiarize myself with the biology. But my first instinct is that this is actually precisely the kind of application this would be very useful for, like looking at things and exploring like molecular mechanisms in disease, and like this CRISPR screening of corrections of mutations or purposeful deleterious introductions, and how that affects neurochemical dynamics, like synaptic variants, probability of release, to extract at a really high granular level of imaging. I think at my first instinct is I think it is possible to do it. In fact, I think this would be the kind of thing this would be very good and useful for. And my understanding is that these are dopaminergic neurons, meaning that we can just use current dopamine sensors effectively to try and do this.
Right, except these are IPS-derived neurons, so.
Yeah, which is and I think that links to the previous question, which is, can we do this in iPSCs versus primary neurons? So these are all primary that I've presented today, and right now we have a collaboration in the lab with a group at MIT that their expertise is in iPSCs, and my expertise is in like making the sensors. And they reached out to us and they wanted to see if these iPSC-derived dopaminergic neurons, like no one has ever visualized dopamine release, like from synapses of iPSC-derived dopamine neurons. Like they want to see that. They want to see if that type of imaging is actually even possible. So I think the first order of business is to really answer that question, which is, from for an iPSC, if you culture it and keep it healthy and happy and then begin to image, would you be able to see the type of these chemical effluxes that we see from primary neurons? And if the answer is yes, that these are like the cellular machinery is all intact and present in iPSCs and it is functional and they are able to release dopamine, then you can begin to ask that secondary and tertiary question around comparing primary specific mutations and that kind of thing.
Would you be interested in potentially collaborating? I don't know if that's an interest of yours.
I have an interest in deploying this tool broadly. Yes. If I don't know, Ilya, I don't know your I'm trying to look at your profile. I don't know if I will be able to get information, but why don't you?
Well, if you
why don't we talk? Why don't we talk by email?
Okay. Could you put your email in your description and I'll just message you?
Okay. How do I do that?
Or I can just send you, Ilya. Just
you
DM
just
me and I'll send you Abram's email address. How about that?
That would be awesome. Thank you.
Okay, very good. Very good. Okay.
I had a question. You had mentioned that you had developed the sensor. I was curious what the technical limitations and potential workarounds for that are of the 10 hertz imaging limit right now.
The 10 hertz imaging, the frame rate was set based on the effectively how bright these sensors are and the signal to noise ratio that is determined by how fast you image them. So if you imagine like a very, very, very bright dye, then you can image it faster, meaning that you can image it like a 10 millisecond exposure, for example, because there is a lot of photon coming from that dye, and there is a lot of signal to noise that you can expand by speeding things up. So we chose 10 because we felt that it was fast enough without actually degrading the signal to noise ratio of imaging. Now we have in the lab developed versions of this that are bright enough to do, say, 25 frames per second, 30 frames per second. We've done that, and it seems to, although it's not our primary choice of sensor, it does seem to work from first impressions. It seems to be functional and capable of capturing activity at that faster frame rate. So that is the reason we chose 10 frames per second of imaging.
Understood. Yeah, there's always that sweet spot between signal to noise ratio. Huh?
Yes, exactly. Yep.
Yeah, does anyone else have one last question? Because I think Abraham probably has to move on to, his research and his work.
Yeah, Katarina, I feel terrible about the technical issues, so I'm actually happy to stay behind, and if there is more appetite for more discussions, I'm available. I don't have anything on the back end, so, and again, this is first time for me using Clubhouse. I've never used it before, and that was primarily the reason I had these issues.
Oh yeah, you're fine. It happens. Like it happens to all of us sometimes that our connection is not perfect or something doesn't work. So, I mean, but it was worth the wait. Definitely because, yeah, it's really fascinating. Also, that you found that with your technology, we found that dendrites also release dopamine. Like that was probably really surprising for you. So did you also then fix those neurons, and is there something different, like are you planning on maybe doing some RNA-seq or something that we could maybe detect why those
Yeah. Oh no, that's you're really poking at something that is very close to my heart, which is to look at this coupling this like synaptically or subcellularly resolved activity, and then mapping that onto molecular level differences. Like, can you imagine taking this, say you took a look at a dendrite of a dopamine neuron, and you have like specific activities coming from specific regions, and then you can go and ask, what are some of the RNA transcripts that are available here but not over there, or protein level differences. I think would be a really cool thing to try and do that. And then if there are some cell level activity differences that you see, if maybe some neurons are more capable of releasing from their dendrites, whereas some other ones remain more axonal in their release profile. Can we couple that with some kind of patch-seq or RNA-seq, right? So where you can patch a neuron and then extract and then sequence it and map these differences, imaging differences, onto transcriptomic level differences. I think it's a really fascinating set of questions that we can pursue. And I wish, and it's all about bandwidth, really. Right now, I have a lab of 2 people, me included, 3, right? So how many of these things can we pursue in a reasonably robust way? So, but yeah, definitely some really fascinating questions that we can go after. Yeah.
Yeah, I understand the trouble of having just so many hands and grants also to do everything we are curious about. I was thinking there was a lab at NYU that did this 3D in situ where you could take the fixed cells and then basically you could reuse and tissue. You could reuse it again and again and then go back and check like other targets that you didn't think of before. But I don't have the details in my mind. It's a few years ago, but that would maybe be cool to do that afterwards, to use their technique. I think you can get a kit from them and just fix it, and then you can go again and again and look for different targets. I think there's just a specific way you have to fix the tissue. So I'll look that up, but that would be really interesting if you could maybe become, I don't know how the resolution would be if you could look at the dendrites versus,
Yeah.
I have to look that up, but that would be
Okay. Okay. Well, thanks for sharing that. Thanks. Yeah, that sounds very. Yeah, the going back and looking at things in multiple rounds of uh, um, looking at for specific molecules. I think that really it does facilitate things. It would be you can do things more quickly that way.
Yeah, the problem is always with dendrites, also patching them or sucking them up. They're so tiny.
Yeah, yeah.
How about, did you try the expansion stuff from Ed Boyden to expand the neurons and then you have more
Yeah,
space?
yeah. Like the person that was primarily responsible for that first paper, that first expansion microscopy paper, he is here. He's at Janelia, and we've been talking. I've been talking to him as well. To like you mentioned, expanding, looking at things. You can also couple this expansion microscopy with multi-round RNA fish where you can image, strip, and image and strip. Yeah, they're all I think potential avenues we can go down. We can pursue. Which one is going to really materialize is still like a matter of question. I think we're going to try a few things and see if which one pans out, which one works out for us.
Yeah, well, we wish you a lot of more hands and more funding so you can do all of it, because I think it's also really important that we just know better how dopamine circuitry works and the release works, and so we can maybe one day solve some more questions around dopamine and
Okay.
everything related to it.
All right. Cool. Very good.
Does anyone else have a last question? Let me check the chat. Yeah, I don't.
Elizabeth is saying Bassoon. Yeah, Bassoon, definitely. The first primary target. Yeah.
Elizabeth, I invited you up to the stage. So, oh, there you go. Hey, how are you? Go ahead.
Thank you. No, is the concept of bassoon really important? If so, could you please tell us what it is? Because I looked it up and it's a reed instrument.
Oh, no. Bassoon is a type of protein that we believe might be playing a role in orchestrating some of these releases. So that conclusion was really sort of like a very first or early demonstration of the utility of the tool, where we're looking at specific proteins that might be enriched or amplified in these regions where we see activity. So, but Bassoon. So if you look for Bassoon protein and instead of just put Bassoon, you will you will be able to find out the function of that particular protein.
Thank you.
Yeah.
Okay, great. I think I could probably go on, but I'm not sure how interesting it is for the audience. But thank you so much, everyone, for coming and presenting your really amazing research. As I said, we wish you all the funding and a lot, that you grow your lab to do a lot of more projects. And let me know if you one day figure out the potassium issue.
Okay, I'll keep that in mind, Katarina. Well, thanks for the invite. I really appreciate the opportunity to come and discuss and talk about this.
Yeah, thank you. And if you one day you have maybe updates, something new you want to share, please feel always invited to come back here. And yeah, I hope your collaboration with Ilya works out. And thanks everyone for coming. Thanks for asking questions and being here. If you like discussions like this, follow the club. We'll have tomorrow Dr. Ilhaik. He will be talking about how he is re-evaluating current bias population genetics. It was a really interesting paper that came out, and actually this paper was mentioned to me by Manas. Thank you, Manas. So she's moderating tomorrow with me. And then my friend Dr. Oliveira Maya will be coming. He will talk about goal-directed and habitual actions, but also about other research he's doing. And Lisbon, he's the director of the Chapalimau psychiatry unit. And and we'll have more rooms. Check out the club. And okay, thank you, Abraham.
Okay, well, thank you. Bye-bye.
Bye, everyone. I'll close the room.
Thank you.
Thank you. Bye.
3, 2, 1. Bye, everyone. Thank you.